This is from the American TV series, The X-Files, which also sought to uncover truths, but in a slightly more fantastical manner. But a lot of the show's basis was based in science and based on unusual observations taken somewhat to the extreme. Now, my office doesn't quite look as tidy as that, in all honesty, I would say. I want to believe, I think is a nice one. The truth is certainly out there. But this is effectively what my sort of lab looks like. And do you know what? I'm not even as honest as this. This is after a lab tidy, and this is after about 2 or 3 years ago. My lab is a lot more cluttered now, and it's starting to resemble a shed, but I'm trying not to get that far. There is still some workable area, and it's mainly just because I keep on acquiring pieces of a kit, not necessarily through the most conventional means. There are all sorts of interesting American Air Force bases just outside of Milton Keynes. It has all sorts of interesting kit that I quite often visit. But effectively, what we're interested in doing is asking and using physical sciences to ask questions about the natural world. And we do that with a variety of different techniques. I'm a biologist by training. I did biological science as an undergraduate, but fell my way into engineering and material science as my professional career developed, mainly because there is something visceral, something I really enjoy about building something, testing something that's not been tested before, and ideally only testing it about 3 times before I get the answer, because I hate statistics. It's really not up my street. My idea of statistics is very politely asking a student to go and read up about how to do appropriate statistics. And that's one of the reasons why I'm not an ecologist. But effectively, what we have the capability of doing within the group is to study the material properties of everything from liquids through to composites using rheology, tensile testing, mechanical testing, then spectroscopic analysis, as well as sort of thermal analysis as well. And we'll touch on elements of those. So whether you have sort of a classical material science background, a bit of biology, a bit of chemistry, a bit of physics, hopefully there's something in this talk that will resonate and find interest for you. And I'm more than happy to hang around at the end and take sort of any questions. We're going to start off with some of these unusual cases, and there's not necessarily a perfect thread, like when I give my silk talk, that sort of connects all of these, because they are somewhat disconnected in their very nature. But the testing of these materials is something that sort of tries to draw them together. And so we'll start off with just a series of sort of questions. And one of these questions started off from some biomedical research that we were doing. We were trying to test rotator cuff tendons. This is your classic ski injury, quite often a tennis injury. If you tear your rotator cuff, you're not able to lift your arm up too much. And I was working with a surgeon trying to characterize the different types of damage that would occur there. And surgeons classify these damage to the rotator cuffs as small, medium, large, and massive. We thought we can do better than that. Surely there's a material property that we can test. The problem with small, soft, and squishy samples is that they don't lend themselves very well to be putting in a tensile tester, because they're soft and squishy and they pull apart and slip in the grip. And that's an absolute nightmare. But effectively what we realized is that you could use a rheometer, which is effectively a cake mixer with a current meter on it, which is a way of testing stress and strain in a rotational manner, or more precisely in a oscillatory manner. So the stiffness. And if you squish a rotator cuff between two metal plates and oscillate the top relative to the bottom, you can get its modulus, you can get its shear modulus. And what we effectively found with the rotator cuff study is that you can perfectly map the degree, or the surgeon's classification of the degree of damage or tear in a rotator cuff to its mechanical properties. When we tried to publish this and say, look, we have this marvelous technique, dynamic shear analysis, as a way of quantifying rotator cuff damage, they said this is really, really interesting. Unfortunately, we prefer to use the small, medium, large, and massive as opposed to put a specific number on it, because it's a bit easier for us. And so we had to switch the entire nature of that paper to supporting the observation as opposed to trying to redefine a quantitative measure of how these materials get damaged. But it led us down a path in which we were able to test biological samples. And one of those started to come across along the lines of knees, specifically human knees. And when you start to get arthritis, and I sort of really feel for you if you do, because it's an awful degenerative sort of morbidity in terms of the problems surrounding it. but testing human knee cartilage and understanding how your knees go prior to the necessary ultimate total knee replacement that you need when bone starts to grind against bone is characterizing the cartilage itself. Now, we didn't have an excess of human knees floating around for us to poke inside this instrument, but what we did have was horses and cows. Here we go. So, as legitimate sort of food industry sources, you can get access to cows' knees relatively easy. You can go down the Moor Market or local abattoir and collect those. You can go to a knacker's yard and get horses' knees. That was an interesting experience. My gosh, there I am. And this is what, 12 years ago? Yeah. So I'm relatively fresh-faced at that point. I hadn't had a child since then, so there was less bags under the eyes and sort of weariness associated with me. I went to this sort of horses, knacker's yard effectively, and the guy was just specialised in large animals, and so he was the one that they would call if they had a giraffe that needed putting down and all sorts. So it was quite an experience, to say certainly an X-file. But getting access to these horses' knees and these cows' knees, we were able to use this to look at different species. And we made the connection that horses are effectively joggers, whilst cows are effectively couch potatoes, because they sort of stand around, sit around all day, whereas horses are a bit more active. And the question was, is their knees optimized for their gait, the way in which they walk? So we have joggers as horses, couch potatoes as cows, and we're able to take the knee and mechanically map the surface of that knee to see if the sponge, the cartilage, was optimized in terms of its properties for each part. And by doing effectively a stiffness map over the surface of the femoral head, we were finding that there are clear differences. It's not just sheer thickness and compression can be done in exactly the same test using this instrument. And it's something that's been picked up quite a bit, actually. People use this relatively regularly now to test small biopsy samples, which can be done in order to characterize these differences in tissue properties. And so that's one of those nice examples where you can take an instrument and adapt its use to ask new sets of questions. And within this biological context, we're also able to make further inferences towards human conditions. Now, this one's a relative sort of outlier. I'm going to move on to this as my sort of next example. This is King Juba II. A prince, a king of Numidia, I believe. Quite an interesting chap. He was raised by Julius Caesar, and he had a real fondness for nature. He was up on the Atlas Mountains and he came across this really interesting plant. And this plant itself was a spurge, effectively. A spurge that he sort of wrote about as having some really interesting properties. These properties were so interesting. Here we go. We'll show a little picture of it, I think, in a sec. There we go. Spurge like this on the Atlas Mountains. You'll recognize it as a euphorbia, potentially. Or if you have Christmas, you've got a poinsettia. That's a euphorbia as well. They're characterized by their milky white sap that's inside them. This sap is particularly interesting. They're called euphorbias because King Juba II named it after his physician, Euphorbidæ. And in honor of him, because of the medical applications that this was found to have. Now, we'll get onto those in a bit. But another curious thing, certainly people in sort of smaller sort of Mediterranean and West African islands use this plant to go fishing. They would, certainly in Sardinia, it's called alawai, if I remember correctly, which is specifically called spurging. They would take this plant, they'd mash it between two stones and just throw it into the water. The milky white sap that is leached from it lacerates the gills of the fish, and they all just float to the surface. So it's a very, very low-tech, low-effort way of fishing. And they're basically, these fish sort of flap around on the surface. And they even have a phrase specifically for this, which you look like a spurged fish if you're relatively confused. Now, hopefully I've not got too many spurged fish in the audience so far. But we're interested in the properties of this milky white sap, not from its sort of biochemical, but also from why does the plant have this milky white sap in the first place? Well, the plant sap itself is actually a latex. It is a milky white substance, milky because it has these large sort of latex micelles in it, effectively. And the question is is that latex used to repair the plant, or is it actually used as an antipedant? Now, if we know fish don't like it, is it present purely to keep herbivores off? And now that would make a lot of sense for a plant, or is it a dual use material where we compared it to another type of latex, not from a euphorbia, but from ficus, so Ficus benjamina. There's probably not one in this room, but you normally find ficuses around sort of hotels like this. But we did exactly the same. We adapted the rheometer and we tested its mechanical properties by getting a very thin layer of the sap and squishing it between two plates, letting it dry and looking at the kinetics of it effectively drying. We found that there are very different types of stiffness increases, so modulus at 0.1 hertz as a function of time for these two different classes or genera of plants. So ficus would set really quickly. It would solidify really quickly, whereas euphorbia would set much more slowly over time. It would be more viscous. It would sort of flow a bit more. With that, we can start inferences regarding how they actually go about setting. Is it a vitrification? Is it a solvent evaporation? Or is it actually a curing like a composite? And with that, we are starting to ask real biological questions about the function of the sap or the latex in these materials as they are being produced and used for the plant's perspective. Now, we've obviously used latex for a range of different things. Natural rubber, for example, is a latex. But this is using these material sciences techniques to ask the biological question about how has it evolved and what would it be used for. Now, I also mentioned other examples of what it would be useful from a human perspective. The sap from euphorbia, my goodness, I was reading up, it's like all of those medical sort of ancient materia medici applications. It's pretty much used for everything. There's a cure-all for everything. Everything from burning warts off because it's quite corrosive, from using it to basically avoid the draft in the Second World War. Boys in Sardinia would wipe their eyes with the sap of this and have temporary blindness to avoid having to be recruited into the army. They would also, in Greek times, use it to treat snake bites anywhere on the body, provided you make a little cut into the head and pour some sap into your skull. You're A-OK, apparently. But I don't know whether it just takes your mind off the snake bite, having this corrosive substance poured directly onto your head. This is another weird example that is quite often seen in nature. This isn't one of those classic parasitic fungi that take over ants and convince them to climb up tall blades of grass so they are eaten and their life cycle of the fungi are repeated. These are bits of plants. They're bits of plants attached to a lovely description. This is a swollen-thighed beetle. These specifically pollinia, collections of pollen from orchids. Well, how do they actually go about attaching? That is a really interesting substrate. When you consider chitin, i.e. the exoskeleton of a beetle, is something that could be hairy, it can be wet, it could be undergoing quite strong forces in terms of it flying from location to location. But obviously the plant wants to be able to deliver that payload to another plant. So what about the actual structures? Orchids in themselves are one of the most diverse and interesting sets of pollinators that you pollinating flowers that you'll get. Incredibly varied. Everything from orchids that will make the smell of a dying animal so that will attract flies, to orchids that are actually shaped like female flies to attract mates. All of these clever little strategies are used. And one of the strategies is that if you get your pollinator close to you, you have to make absolutely sure that your pollen gets onto it. Now, that's a really big case for orchids because orchids aren't like most sort of flowering plants that have lots and lots of pollen. They put all their eggs literally into one basket. This pollinia that's all of its pollen, and it detaches once onto its pollinator, and then that's it for the flower. So it doesn't get multiple bites here, it has to attach. And so how does it go about attaching? Well, it attaches via this plate here called a basidia. And then it also is attached to this larger structure, which is known as the pollinia. we were interested in understanding how does it actually go about adhering to substrates. With this, we adapted our tensile tester, and we were interested in understanding, well, what actually happens as we're pulling it apart. So not only did we pop a camera on the side of our tensile tester, we popped a camera underneath our tensile tester and attached this to a glass slide so that we had a contact area measurement constantly as we're undergoing pulling of this. Now I have to play this video, and it's a slow one. It's okay, it's not too far. But the interesting thing surrounding all of this is just how long it can take to start the test. And here we go. We're starting the test. We're pulling this material. We're deforming it. It's exceptionally elastic, this connecting part. We're actually doing it so we're getting a live force reading here. This is a reading straight off the instrument in terms of its mechanical properties whilst we're measuring it. What we're finding is that the actual vicidial plate does not move at all. That contact area is stuck properly onto, admittedly, a glass substrate, but it will be the case for an insect as well. But what the remarkable thing is that the strain to break on this very, very small attachment here, where the Holland is ultimately attached to, is incredibly large. I think we end up getting, okay, we'll get much, much further here, but we effectively have a grip-to-grip separation, i.e. a distance there, of around about 11 millimeters before it breaks. So this is a strain of several thousand% before it ultimately snaps. These are strains that the animal would never encounter in terms of it could be really, really beaten up, and this is not going anywhere. So once adhered, it's incredibly adhesive. And we can start to also measure the mechanical properties of this segment as well as the plate itself and do some comparisons. And what we found is that throughout all of the orchids, not only did they have multiple types of adhesion mechanisms, including one really weird one that we think is thermally adhesive. One is protein-based, one is polysaccharide-based. There's a real richness to the different types of adhesives there. But they match the vast majority of different types of medical adhesives out there, yet it's still a natural material which could be much more compatible in the future to use if we can sort of understand how nature makes adhesives like this, but also some other sort of really impressive biological adhesives. But as with all of these things, the case isn't closed because we've not published the paper yet. It's been submitted, and we're awaiting sort of feedback. But this is kind of the fun of it, of this talk. Moving on to another example is what is the point of a guillemot egg. Now, there is a myth. We can myth bust a little bit in an X-Files talk, I suppose. These are guillemots. They like to spend their time on cliff ledges, mainly because snakes and rats don't like to spend time on cliff ledges as well. But there is a constraint to that in the fact that if they're nesting, they don't really make nests. They just sort of pop their sort of eggs straight onto the hard, rocky surface. And I mean, I don't tend to keep my eggs on hard, rocky surfaces because they're quite fragile. But they're also very interestingly shaped. These guillemot eggs are not your typical sort of ovoid shape. They're quite pointy. Now, one of the thoughts and the reasons why they're quite pointy is that if you were to roll it, it will always roll in a circle. And that's great because if it's on an edge and it starts to roll, it will always roll back towards the cliff. But they did quite a bit of work on that, and they found out that's really the case. The rolling arc of a guillemot egg means it would still roll off effectively. So the theory is that the guillemot eggs stop them rolling off cliff edges. But Prof. Tim Birkhead, who's a biologist over in Animal and Plant Sciences in Sheffield, was a bit unconvinced. So he wanted to understand, well, what is the selection pressure that's acting upon the unusual shape of these things? They're incubated on the rock surface, so there's no nest. And so a postdoc worked with our group a few years ago to test this theory. And so we adapted Arswick once again to a guillemot egg holding device, and we effectively poked it at every single point on the surface to understand what its mechanical properties were, or effectively the stiffness. And what we found is that, yes, there are differences in the mechanical properties across the surface of the egg. What we think is actually going on there is that it's spreading the load so that when the guillemot comes back in, flies back in and tries to incubate, it doesn't break the egg and also provides quite a large surface area for incubation, so that the breast of the animal is fully transferring the heat into the egg. So it's not to do with stopping it from rolling off. As we sort of continue this menagerie of unusual examples, does anyone know what this is? It looks a bit alien. Yeah, it's an echidna. I found out today that all taxonomic examples, all the taxidermy examples of echidnas are wrong, because their back legs are actually on back to front in real life. But all the taxidermists looked at it and thought, well, obviously they're never going to be on back to front. We put them the right way round. So a little bit like the dinosaurs in Hyde Park. When we were recreating them, we got it wrong. But these things are extremely unusual. They're monotremes. So we've got 4 species of echidna, 1 species of platypus out there. They're native to Australia. They have a pouch. They lay an egg. And they are a mammal like you or I. So very, very weird beasts. And on top of that, they are a defensive structure. By the looks of it, they're covered in these spines. And I've got a PhD student who spent the past sort of 3 or so years understanding what really is the point of a spine. And so animals have evolved these specialist hairs, which are called spines. They're called quills if they have a hollow center. And by looking at all these different animals that have spines that are not necessarily related to a single spiny animal. So this is an invention that's happened multiple times in nature. Everything from the porcupines through to the echidnas, these very unusual things called tenrecs, and the familiar hedgehogs. We know that hedgehogs have spines effectively to cushion themselves because they like to leap off things and roll into a ball. So that's a shock absorbing mechanism. The Cape porcupines and the Malayan porcupines have very, very, very large spines, and they sort of effectively find the nearest hole, burrow in, bum out, and then hope something runs towards it and gets a mouthful of quite horrible porcupine spines. A lot of the literature around porcupine spines is medical literature about how to remove one, because they're also quite like your classic sort of archery and arrows, designed to break off as well. But as we keep on looking more and more and more into them, they have some really unusual features that have not been reported. We have, I think it's one of the, I think it's Malayan porcupine as well. It's effectively shaped a little bit like a saber. It has 2 sort of pointed edges that go down, which will help with the penetration. But the question is, are they for attack, defense, or other? And the only real way that we can find out, apart from actually looking at how the animal uses it, which is quite a tricky thing in the first place, is to ask material questions about it. And also to determine, well, how sharp is a spine? And the other thing we realize is there's not really a very specific definition of what sharpness actually is, or it's certainly a movable feast in terms of how you decide how sharp something is, in terms of if you look at the very tip, is it the angle that it comes into? Do you fit a small sphere onto the end of it to work it out? And do they have specializations effectively for puncture? So once again, we've adapted our tensile tester to use it as a puncturing device. By fitting our spines on, we can look at how they impact and penetrate something like a ballistic gel or something with the modulus and stiffness akin to flesh. We've actually used a chicken breast at 37 degrees C to test it properly, because that is the kind of material that it's going into. But we can measure the forces going in and also the forces required to retract. Is it harder to retract to make sure that it breaks off or detaches from the animal itself? But on top of that, what we've started to do as well is that we've been combining this with some polarized light imagery as well to look at deformation of the gel, to understand how the stress is distributed, in order to see, is this a focusing the stress in one place, or is it spreading it out? Now, that will determine the type of pain that is perceived by the animal and the pain receptors that will be activated, but also could lead us to understand how we could make pain less or less painful needles from injection purposes. So there's lots to learn about the natural world. We're moving on to other sort of hairy rodents. This one is sort of one of my favorites in terms of the story surrounding this one. This is Lophiomus imhausi, found around sort of Western Africa or so. But it's a rat. It's only about 14 inches long, 35 centimeters long. But it's quite unusual in the fact that it's been well known to kill dogs. So if dogs try and attack it, they come away much for the worse, and about a day or so later they die, which is very unusual for something that you'd consider having that as a pet potentially. So we were very interested in understanding what's going on there. And this fantastic naturalist called Jonathan Kindon, who literally wrote the book, well, the 7-volume tome called Mammals of Africa, exactly the kind of person you would expect to be running around Africa, making detailed drawings and anatomical descriptions of all of the mammals of Africa. And he came across something quite unusual. When updating his book, he was going through his library of materials, and he came across a couple of the skins from these Lophiomus. And unfortunately, the moths had got to them. And the moths had got to them and eaten the hairs everywhere, apart from a very specific strip down there. And that strip is here. And all the hairs were eaten apart from those two bits. So he thought and came to us and said, basically, there's something weird going on with these hairs. Can you have a look at it? Well, we just put our material science hats on. First thing we did is we put them under the SCM. And under the SCM, these are the most fascinating structures. This is hair. This is hair like on your head hair. But they are expanded. They have all these little fibrillar structures inside, but on top of that, they also have all these big holes along them, which strikes me as being quite unusual. You don't tend to want holy hair. So we took SEMs of it, but on top of that, we also subjected it to some FDR, some chemical analysis, and we found out that it had uabane in it. Now, this is quite an unusual toxin. It's a toxin that the animal doesn't produce. And what we found out is that if we look at the structure of this hair, and as an example, it does this really unusual case of wicking. So you put a liquid on it and it will wick and fill the contents of that hair. So what's going on? Well, this is an example of the world's only poisonous mammal. Now, platypuses are venomous, as in if it bites you, you die. This is if you bite it, you die. Okay, that's poisonous. That's the difference between poisonous and venomous, depending on who's biting who, effectively. But this hairs seem exceptionally specialized. And the answer to this mystery was actually in the original video. This animal does is that it chews the bark and roots of the poison arrow tree, sequesters the poison into its saliva, and then turns around and licks these specialized hairs, wicking the plant's toxin into it, so that the next time a dog decides to try and have a go at it and bite it, it will get the sequestered poison into its body. And that's something that we find through material science approach to understanding what's going on here. Absolutely fascinating beast, to say the very least. But we've moved from poisonous. We're going to spend a bit of time talking about venomous now. But within venoms, and as we'll get to talking about sort of my main area of research, silks, I'd like to introduce a new concept to you. And this concept is a concept of a material called an ectosecretion. Now, a lot of the stuff that we've seen so far tend to be materials that are used inside the body. For example, the plant using its sap to heal itself, or the cartilage of a horse or a cow. But ectosecretions are a term of materials that is a material produced by an organism, but specifically for use outside of the body. Now, there aren't many ectosecretions, true ectosecretions. You can start to sort of think of them: venom, spit, mucus, silk. These are materials that have evolved to work outside of the animal. Come wind, rain, sun, shine, they perform a function and extend the physical manifestation or impact of that animal in the world, its phenotype, outside of its body. which is great because as material scientists, we use materials outside our body. We are interested in how materials can perform outside of our body in a range of different circumstances. And ectosecretions work at the extremes. Yeah, it's not nice, easy, wet, easy to regrow, heal. Once it's outside the body, it's got to deliver. Otherwise, that's it. The animal gets eaten. So ectosecretions are an area that we've spent quite a bit of time sort of focused on and looking at. One of them is this. Here we go. Not a very happy chap there. Snake venom. And knowing what we know, which is we are very good at testing small amounts of squishy samples, one of our questions was simply, is there a difference in the venom between a spitting and a non-spitting cobra? Now, that's an example of a cobra spitting. It's quite cool, actually. It's twisting. It actually goes cross-fanged, effectively. And they're so agile, they can actually direct each fang in different directions, aiming for the eyes specifically, in order to deliver their payload. But is that protein solution, that material that's used outside of the body, optimized for its delivery mechanism, i.e. a fang? Effectively, what we're looking at here is, are you able to push at high rates a protein down a long, thin tube and make sure that it still works by the end of it? And that's exactly some of the problems you have with vaccines, and not beating up these very expensive biological molecules and ensuring that they are able to be processed at high rate, at high shear rates through small orifices. Can we learn something by the way in which nature has solved exactly the same problem by looking at the material properties? So that's effectively what we did. We looked at snake venom. We didn't do this. Our lovely friends at the Liverpool School of Tropical Medicine very kindly milked dozens of different tropical snakes for us. I mean, glad it's them, not me. My goodness. We just had a very, very lovely chap called Ignacio turn up on the train from Liverpool with a bag of little vials that we were able to pop straight onto the rheometer. And we did that. There's Ignacio there. He's not as experienced. He's got the back end of the snake. But effectively, we popped it onto the rheometer and we tested the difference between effectively spitters, non-spitters, and an out group to understand, are their flow properties optimized, depending whether they spit or not. And do you know what? The case was never closed. You can't tell a difference. We don't know why. There is no real big difference between spitting and non-spitting cobra venom, which is frustrating, to be honest. It made it for a very, very difficult paper to publish because there was no result. But we were thorough, at least, and we compared it to doing pressure predictions down fangs that had actually been CT scanned, knowing the length of the tube, and measuring the pressure required to do it. The pressure is quite high in spitters. It's about sort of 10 atmospheres, 100 meters below sea level. It's quite high forces that it's subjecting it to. But we were unable to really close this one in terms of, is there a difference between spitting and non-spitting cobras? Because we couldn't see much of a difference between them. But that's part of the fun of life, isn't it? On to another ectosecretion. I've obviously found lots of videos of snails, but I wanted this one to be just quite sort of uneasy, because it is, I know, it sort of makes you feel, oh, I'm not too sure about this. I mean, people pay for this. Let's just think about that for a second. People pay for this treatment. But I'm going to reassure the people by the end of this that it's not as bad as you potentially think. So we were interested in understanding how snails and slugs move. Now, a few years ago, we had a fantastic entrant from this chapter as well. Lauren Egleton talking about young person's lecture competition, was a UK finalist as well. Understanding how do slugs and snails move. Now she's got her PhD. She's now Dr. Egleton, and this is some of the work that she did just at the very end of her doctoral studies. And so while she has understood some of the material properties of the mucus, one of the questions that we could never really work out is how thick is the layer of mucus that comes out of a slug or a snail, partly because we didn't really have the tools in order to measure it. Well, this is one of the tools to measure it. We found a very nice chap called Steve Matcher, who works in EEE, so electrical and electromechanical engineering, and he has a technique called OCT. Now, if any of you have had an eye test and decided to pay the extra £5 for a nice picture on the back of your eye, that is exactly the tool that we were using. So optical tomography. It is a long working distance, three-dimensional imaging line scan, effectively, of different optical densities or phases of a material. So we popped a white light one here, as opposed to an infrared one that tends to look at the back of your eye, and we ran a slug over it. So we got a nice little video here of them pootling along. This is the glass substrate here, and now you can see the slug as it's moving over, and we're able to measure the distance between the slug and the substrate. On top of that, you can start to see these pulsatile waves moving in because it's actually inside the slug now. So this is looking at its muscles. And we can see the deformation, the height. We're starting to get stresses, strains, information about how this mucus is deformed as the animal is using it. And importantly for our group, without messing around with the animal. Yeah, we're not scraping it up afterwards, rehydrating it. This is the material in its natural context as it's meant to be naturally used. So this is always something that we're particularly sort of interested in understanding, and it's going to get towards the end of its journey in a second. And you can really start to see these waves now coming along. Now, as we've got this as a function of time, we're able to plot that distance along the animal, function of time, for thickness of the mucus. And there have been hypotheses and there have been suggestions about the thickness of mucus in literature in the past, but this is one of the first times that we've been able to actually measure it in use. And there we go, it's popped off now. And what we tend to find is that over time, it's pretty constant, the depth of mucus. And on top of that, the mucus is actually a lot thinner than we had previously expected. We're only talking sort of, 5 to 10-ish, maybe 5 or 6 microns thick, which is thin, very, very thin. On top of that, what we were trying to understand is, well, where does the mucus come from? Is the mucus always sort of pumped out across the whole of the foot like a hovercraft? Well, that wouldn't really make much sense, because if it was constantly coming out at a constant rate all over the foot, the back end would be higher than the front end because it would be constantly accumulating as it's going forwards, but it was quite flat all the way, which means that the mucus has to come from somewhere else. And so Lauren has actually just recently found where the mucus comes from for locomotion. It's just under the chin. It's got this massive suprapedal gland that no one had ever really associated. What does this big thing do? In all gastropods, snails and slugs, and it looks like it just sort of dribbles constantly from under its chin as it glides over it. And on top of that, we found out that they never touch the ground. So they ice skate. They're always riding on a layer of mucus. So hopefully, to try and make that person who's got snails on her face feel a little bit better, at least she knows they're not actually touching her face. They're just sort of like gliding over a bit of mucus. Right, so how long do I have left, actually? If I... Yeah, sure, cool. Okay, well, I'll go into this then. This is sort of an origin story. It's always nice to have a little bit of an origins story. This is actually my favorite episode of The X-Files. My goodness, I loved it. Darkness Falls, season 1, episode 20. It came out 9th of February 1995 on BBC Two. So that made me feel suitably old as I was Googling about an hour and a half ago, when did this first come out? So 31 years ago, this episode aired. And it's an interesting episode, not because it has a specific like ghoul or ghost or alien attached to it. It is a story about how a group of loggers go missing in the Vancouver forest, I believe. And Mulder on the left and Scully on the right, along with the park ranger, go out looking for them. They find a lot more than they were expecting. They find an eco-terrorist, but they also find these things up in the trees. Silky cocoons. silkiness. Now, whether this was one of these subliminal things that lodged into my mind, activating 15 years later when I'm trying to determine what on earth I'm going to do for the rest of my academic career, but this cocoon was cocooning up people. So this animal was effectively cocooning up these loggers. Turns out in the episode, it's a series of little aphids that were light sensitive but would swarm and create silk around people as they sucked them dry, which is not unlike some of the animals that also produce silk. We know that spiders, for example, effectively digest outside of their body and then suck in a soup. Spiders don't have teeth. They actually have a little filter. So they excrete all of their enzymes and then just suck a nutrient soup into them, which sounds horrific, and it relatively is. But it was the material that I started getting interested in. And so some of the stories about the other functions of the material, we profess to how amazing silk is in terms of its overall toughness and its fantastic, or relatively fantastic strength compared to its density. But when you consider what silk has to do for a spider, it has to do much more than that. This was one of the most interesting PhD projects that I probably supervised. It was, she's now a prof in Hertford College in Oxford, Beth Mortimer, working on vibrometry, effectively, or bioseismology, I think she calls it, which is effectively understanding how vibrations are transmitted in nature. And her PhD was effectively to fire bullets and lasers at silk and understand the mechanical properties of them, because understanding how silk responds at high rate deformation can help us understand scenarios when we're requiring them to, oh, well, we're not going to get a video of that, unfortunately, how they can be dissipated, for example, when an aerial insect hits a web, but it can also be used once they hit the web as an information gathering technique. It's remote sensing. Spiders can't see very well at all. They live their life through vibrations, and they feel where the animal is based on vibrations on their web. So a web is not only an arresting device for aerial impact, it's also a remote sensing device. So looking at how these materials interact under deformations at the rates for sonic propagation is exactly what Beth did. Now, this is the fanciful sort of spider silk bulletproof vest, which I've mentioned before. It doesn't work. It would stop the bullet a couple of meters behind you, but you'd have to pull it through the hole, because as we all know, things can be tough, but at the cost of deformation. But what she was effectively doing is that using a sort of 140,000 frame a second camera, and I bet this won't work just to spite me, no, it won't. We were doing a streak imaging of deformation under high rate. And when you hit things that fast, the thing that determines the mechanical properties is effectively the speed of sound within that property, within that material. And what we found with spider silk is that it has one of the highest, widest ranges of tunable frequencies in transverse propagation of sound compared to any material. And the spider uses that. When it's making its web, it tunes its web. It plucks it. It applies tension to the individual threads so that they will transmit the right sound in the right direction so that it can listen to prey when it hits it. Another example of that sort of comes along the lines of this. Now, I used to look after a greenhouse full of spiders, a couple of hundred spiders out in an old sort of 1920s, 1930s wooden greenhouse. And they were very much free range. They were just allowed to sort of go wherever they wanted. And it was my job as a PhD student to cycle about 6 kilometers out there every single week with a bucket full of fruit on my back and a bag full of flies and a brush and basically clean out and feed them, which was okay. It kept me fit, I suppose. It wasn't really great in the wintertime when it was dark in the evenings about 3 or 4 o'clock, because I had to use my bike headlights to go into this thing and make sure I didn't sort of walk into any spiders. In the summertime, when I give it a proper clean out, I opened a door one day and a bird flew in. So the bird wasn't harmed, the spider wasn't harmed, but I did take the liberty of taking a very quick video of this bird trapped in a web. And when you consider the forces that this bird can apply to the structure, it wants to get out. It is completely arrested by an animal that big with a structure that you cannot see. it has evolved to be invisible. So this is a perfect example of the amazing mechanical properties and the energy dissipation mechanisms provided by silk. So we then started to think, well, what about invisible silk fibers? If silk fibers have to get smaller and smaller and smaller, is there a lower limit? And what happens when silks get really, really thin? Well, the best place to look for that is you look at an adult spider and it doesn't really sort of help you because its dragline silk is about 5 microns. So it's around about sort of a fifth to a tenth the width of a human hair. And you can still see those, they're manageable. When you're trying to load them onto a tensile tester, you can catch them in the light, you can get a glint, and you can start to sort of see where they are. The real challenge is when you start to look at their babies. So we've never published this. It was more of a technological demo between myself and genuinely the nicest chap I've ever met in the world, called Holger Pfaff, who used to work at Keysight Technologies. I think he now works for HP, but absolutely lovely gent. And he had a very, very expensive piece of equipment, which is a tensile tester that costs a house. It was about £250,000 for this tensile tester. It was effectively 2 AFM transducer heads on top of each other to give a sensitivity that allowed you to do mechanical testing on fibers that you literally couldn't see. The only way I could load this sample into the cardboard frames is to take almost somewhat of an Occam's razor approach. I would have a baby spiderling on my hand and I would drop it off and it would be floating in the wind. And I knew that it didn't have anti-gravity technology. So there must be a piece of silk between here and here. And so if I sort of fished, fortunately, with a cardboard frame, I might be able to catch a single strand of invisible silk between it, popping it onto that instrument. And this is the general performance envelope for a dragline silk. And when scaled to the size of it, these things had exactly the same mechanical properties as their adults, as their parents. You know, a classical sort of initial stiffness and then a yield and a bit of work hardening. But the really cool thing about that is this is a of the cardboard frame and the sample. We've got 1 millimeter here. So normally you'd just about be able to see this, but we have to zoom in a little bit more. So it's 100 microns. We're starting to see the fiber now. And here we go. So this is full property testing on a 225 nanometer individual fiber. So no wonder you can't see it because light doesn't interact with it in the visible spectrum. It's as simple as that. You literally couldn't see this thing. But it just goes to show that nature has the ability to scale its material properties across length scales and yet still deliver what the spider needs, which is a mechanism in that instance for ballooning and potentially making a small spider web. Right, I'm a little bit aware of time and the rest is somewhat sort of silky, so I'll be relatively quick on this last bit because we looked at how things were encapsulated and rapidly sort of wrapped around prey as a spider, but there's another example of prey or wrapping for protection, which is a silkworm cocoon. This is where we get all of our commercial silk from. And it comes from these things. So not a spider, but a silkworm. This is a caterpillar, effectively, that produces over 1 kilometer of silk in a cocoon that is easy to dye, easy to unravel, because we have determined it to be so, because we have agriculturally reared these animals for about 5,000 or 6,000 years to be one of the world's oldest commercial fibers other than wool. But the unusual question that we asked is, well, is silk pushed or pulled out of the animal? Now, most people think, well, is it squeezed like a tube of toothpaste? Well, maybe. So we wanted to use simulation and experimental validation to determine if silk is pushed or pulled from the animal. When people have done previous measurements using artificial spinning, they would refer to the pressure and the overall spinning speed, and the pressures have to be quite high. This led to one of my favorite diagrams I've ever done in terms of research, because this is the range of spinning speeds that the animal naturally sort of occurs, and it's around about sort of 1 to 100 centimeters a second, effectively. And if you force reel it, as in pull silk from the animal, you can go very slow. You can go sort of microns a second up to practically smoke coming out of the back of the spider at sort of 100 centimeters a second. But for all of those things, the pressure has an upper limit, and that's the rupture pressure. This is the maximum internal pressure measured not by us blowing up spiders or silkworms, but the pressure that's required and measured to burst out of a chrysalis. And so they've done measurements on that we took as, well, this is the upper limit that it could produce. And what you see is that even if the animal was squeezing as hard as it possibly could, it would still be 4 orders of magnitude lower in terms of the pressure that it could generate internally. And so we thought, well, that's it, really. There's no way it could possibly be squeezed. But unfortunately, the reviewers said, well, that's all very nice as simulation. But any chance you could show that experimentally? And we changed everything from duct geometry, slip, viscosity, and it can't enter the pressure requirements for natural spinning. And experimental validation agrees with it. The pressure to push is too high. So finally, we took our Zwick, popped unspun silk into a syringe. We had a syringe that was the same diameter and length as a natural silk duck, and we tried to push it out. And it turned out that it did have a huge amount of force required to do it, and it matched our predictions. We also were able to measure the forces required to pull silk out of the animal. And that was by taking a silkworm and thinking that a rheometer is just effectively a fancy cake mixer with an amp meter on it. We did exactly that. We turned it into a spinning device. We turned it into a mandrel. We set a constant speed of rotation for the rheometer, exceptionally sensitive force measurements, and measured the forces required to pull it from the animal. And the forces required to pull it from the animal are perfectly possible by the animal. It doesn't really require much to pull silk from the animal, but it requires a huge amount to push. Right. So that leaves me towards the end of the talk. The findings, I don't know whether the truth is out there or whether I want to believe, but I'm going to leave us with, as quite often happened in the episodes, a quote from people who are much wiser and more experienced than us in the past. And this is from Monsieur Le Bon, who was president of the Royal Society for, I believe it was science in Montpellier, but also the mayor of Montpellier 300 years ago, who wrote a discourse on the usefulness of silken spiders in 1752. He wrote, so many examples ought to show us of what importance it is to neglect something in the study of nature. What at first seems of no use or almost impossible to be put into execution, oftentimes turns to the greatest advantage and becomes easy by care and industry. And I think that's something that I'd like to take away, is that if we can look to nature, we can find solutions. And if we can implement those solutions, they will often be a more economic and usable solution to the ones that we currently have. So with that, I'd like to thank you all for your time. This is my current group at the moment, and I'm happy to take any questions. It's a really great question. So there's an honest answer. I'm just fascinated by nature. Yeah, I'm just really interested in the unusual use cases. And then there is the practical current funding climate answer with, yes, got an absolute plan for an application of that. There's definitely a route to market and a commercialization sort of pathway planned out. And I think we're going to be forced more towards that sort of way of thinking in the future as a sort of act of necessity. But my honest feeling is that the best discoveries and the innovations have come Friday afternoon experiments. You know, we've got things that have been bubbling under for years. I've got quite an interest in spittle bugs or cuckoo spit that we see, but we only get that once a year for about 2 or 3 weeks in sort of April, May. And so I've been slowly building up enough evidence to really try and understand what's going on there, because that could be an amazing biological foaming agent. If you could have the head of your beer last around for a couple of weeks, come wind, rain, sunshine, like these things do outside, it'd be amazing if we can work out those kind of surfactants. But you've got to capture it. You've got to try and get access to those materials. And I think it's access to materials is one, and just a general curiosity that really drives me. So. So I think the 2 electrons would be, we use, and they're standard SEM, not TEM. So scanning electrons were for the the hair from Lophiomus, as well as the baby spiderling. So, but the other stuff tends to be optical. So I don't have an EM in my lab, but I do have optical I'm quite obsessed with optical microscopes. I love them. I collect them actually at home. I collect Vickers microscopes, partly because I used to live in York and they were made in York. Unfortunately, I don't have much uptake from my students on optical microscopy. You look at my papers, and we don't have many optical microscope images in them, which I feel is a real shame because I love it, but I will eventually wear them down. Almost, but there's this amazing academic called Connie Rodenberg who has worked out color SEM recently. And that is so cool and a really clever idea that she basically invented. If you look at backscatter electrons and you're firing electrons into your surface, you've got your detector and you're measuring how they're bouncing back. Well, what she worked out is that you can effectively, and how high they bounce is related to their energy. If you filter or move the detector up and down, you can look at how high they bounce, which means that you can get a spectra of energy absorbance of the electrons for every single pixel or location, and you can convert that spectra into chemical information. So you can avoid density issues because you can map that out, but you can effectively do chemical imaging using standard electron microscopy, which is flipping brilliant. Honestly, so she calls it CHIMES, I think, color hyperspectral imaging. It's really cool. You can see disulfide bonds, you can see different protein conformations. They've used it for it's all sorts of harder materials as well as the soft materials. So we are getting there for color imaging in SEM, but it's not quite the color that we think of it. Yeah.
Thank you. There's one over here.
Yes. Mm-hmm. I have to remember this, to be perfectly honest. So the cows were a bit more stiff and thin because they're not loading it and it's not having impact loading. It doesn't need to cushion, whereas the horses tended to be a bit thicker and a bit more squishy, if I remember correctly. Yes. Yeah.
That sounds like personal interest there. There you go.
I found out recently, I found out only yesterday from Keith Jackson, who's a master cutler at the moment, but he used to sell operating theatre solutions as part of Brandon Medical. He was telling me yesterday to one of the courses that I run to the students, they have operating theatres just for camels, just for racing camels. So expensive are these racing camels, they can dedicate a whole operating theatre to one camel to make sure that it doesn't knacker its knees. Isn't that amazing? So there we go.